So, happy Monday. We are coming to the last week of classes, and I know you guys had that paper that was due just a short while ago. You have some discussion questions due tonight at midnight. Please don't forget to do those. Those will count as your attendance points. So, for those, I really just care that you thought about it and you answered the questions to the best of your ability, not that you got them completely correct. I won't take off points if it's not the perfect answer that I'm looking for. I really just want to see that you've answered all of them and answered them with more than just like a one word answer. So you've like thought about it, okay? Like a discussion. Okay. If there are questions about that, you can chat. I'm going to jump to our PowerPoint. Okay, let's see here. Okay, wonderful. So, we had left off last week with a scary slide. So we had left off with this slide, right? Like all the things. And it is scary. If you break it down and you follow each arrow and you kind of talk it out to yourself, it does make sense. You know, I like to start actually like right here, following this middle line, you know, thinking about what happens in times of dehydration or loss of blood pressure or blood volume, and what happens in the kidneys, specifically with those juxtaglomerular cells. They're releasing renin, which is going to be involved with that whole chain from angiotensin 1 to angiotensin 2. And then angiotensin 2 does a few different things, right? Releases ADH, otherwise known as vasopressin, causes the release of that from the pituitary, causes the release of aldosterone, right? You know where that comes from, what those specifically do in the kidney, what effect that has, right? As well as the effect of our sympathetic system, and other things helping with vasoconstriction, all of this coming together to lead to an increase in blood volume and an increase in blood pressure. So I know it's a lot. You guys drew me some pictures of the RAS system. Most of them were great. A few people needed to provide like a little bit more details. I know this slide is overwhelming, but this is kind of like the level of detail we're looking for. Like we want to know not just that, for example, not just that renin is coming from the kidney, but what cells release that. And not just that it affects the kidney in the long run, like at the end of the line, the kidney is going to be affected, but where specifically are aldosterone and ADH, otherwise known as vasopressin, where are they acting? What are they doing? And so that's kind of the level of detail that I expect from you guys with regards to this material. So yeah, that's to keep to kind of direct your studying. You got to know it like that. You got to know it like details. Okay. If you have questions. Okay. We're gonna now move to kind of our last part of the PowerPoint, and comparative anatomy. So this, which you covered on your own. The way I like to structure things is a mix of me giving material with you finding information for yourself, and then reinforcing it. The more times that you actively engage with material, the more you're going to learn it, and the more sticky it's going to be in your brain. So that is kind of the strategy behind me having you work on stuff ahead of time and then me kind of come back around to it, if you would. So our learning objective here is to talk about the kidney and the different structures that some animals use. Again, I'm not going to cover it in wide depth because you yourself already read about it and did some work with it, answered a homework about it. And so this should be more of a review, but invertebrates use that primitive kidney, that protonephridia. In worms, they use that tubule. It's got a pore on one end for a filter, and with a cap cell, and on the other, we've got a propeller, like a flagella or a flame cell. This is what that's showing, right? We've got that flagella here. As we move through evolution, we have in the mollusks a metanephridia, which is, as you can see, it's a little bit more complicated. It's got some invaginations here, and it's longer. The length has to do with how much need they have to expel water. So it's longer in animals that need to expel more water. As we move through evolution, we get insects, and they've got no single tissue that acts like a kidney. Instead, they use two different tissues. So they've got a Malpighian tubule, an MT, and that empties into a structure called the hindgut. And the MT will vary between species. It's more complex in those that eat things with a lot of water in them. Think blood and mosquitoes, or the insect eats a lot of sap. They have a lot more liquid. And in this system, there's no direct filtration, but instead they modify the primary urine. So it all is one fluid that's then modified. But it can be acted on by hormones. And some of the hormones that we talked about were first identified, or homologs of those hormones were first identified in insects because they have these principal cells which can transport solutes, and they've got these stellate cells that have aquaporins and that have water. And so they have this separation between transporting solutes and transporting waters, and there's hormones that act on them. And so it was a system that we use a lot when we want to learn about the effects of different drugs, for example, or hormones. We can use things like Drosophila and other small insects to learn about that. As we move into fish, keep in mind, again, whenever we talk about fish, there's a difference between freshwater fish and marine fish. So in freshwater fish, they've got a larger glomerulus and a larger distal tubule. That's shown here, and you can see that because they need to recover more salt, right? They're in a less salty environment. They need to recover that salt, and they're going to use large volumes of urine. Whereas marine fish, there's a wide array of ways that marine fish have their kidneys set up. In general, they make less urine and they have a shorter proximal tubule. And in general, or there are some of them that have no distal tubule, and some also have no glomerulus. So it depends. There's a wide array. It's hard to say, but again, you want to keep in mind the goals for freshwater fish. They're trying to recover more salts, and for marine fish, their goal is more on recovering water because they have so much salt. They're trying to expel that salt. Okay. Amphibians, interestingly, use their bladder as like a water storage pouch with them. So when they're in water, they can just get rid of excess water. But when they're on land, they need to conserve water, and they don't have a loop of Henle to help make that hyperosmotic urine. So instead, they'll just recover a lot of water from their bladder, and they'll use their bladder as like a water storage tank for when they need it, which is interesting. They drink their pee, kind of. Not really like drink, but they use their urine more effectively. Terrestrial animals, we use kidneys to help us conserve water depending on the environment. So reptiles, they're going to use uric acid as their waste, which again really helps them conserve water. And we talked about that already. Vertebrates, again, this is where we have the structure of the loop of Henle. And you need to really be familiar with the descending versus the ascending, what happens in each. But the loop of Henle really helps us and other vertebrates produce that hyperosmolarity urine, that concentrated urine. And the best predictor of the ability to concentrate urine is medullary thickness. It's the width of the medulla as a fraction over the total width of the kidney, because the loop of Henle spans that width. And so if the loop of Henle takes up more and more space of the total kidney, that's going to help the animal conserve more water. And so you're going to see that medullary thickness be very high in desert animals, because again, they really need to conserve water. And this is what that is showing. So in animals like a beaver, they're in water all the time. They have a shorter loop of Henle because, you know, they don't need to conserve as much water. Rabbits have a mix of both because their environmental needs could change. Whereas a kangaroo rat that's found in the desert, that's going to really need only really long loops because it's always going to be in an environment where it doesn't have a ton of water. Okay. So that is the comparative anatomy. Now I'm going to touch on one disorder, a short disorder, and then a few drugs that affect the kidneys. If we had been meeting in class, we would have had a much longer lecture about drugs, but I'm just going to give you some highlights here. So our learning objectives are to describe diabetes insipidus and differentiate two major forms, nephrogenic and cranial, and then to explain the mechanism of action of key drugs that act on the kidneys and define specifically where in the nephron they act and on what channels or transporters they act on. So diabetes insipidus, the symptoms for this are polyuria, so they're peeing a lot, polydipsia, they're thirsty a lot, dehydration, and low blood pressure, especially when standing. So they might pass out when they stand up. And there are two types depending on what started the problem, but they all relate to ADH, antidiuretic hormone. And basically, there is either a low response to the ADH being made, or there's not enough ADH being made. So in nephrogenic, there is a low response to the body's antidiuretic hormone because of a mutation in the antidiuretic hormone receptor. So the receptor is mutated because of a gene, so it was passed on through the family or happened spontaneously. And so that person is making enough antidiuretic hormone, but their kidney is not responding to that antidiuretic hormone. Again, you need to know where in the kidney antidiuretic hormone exerts its action. Okay. Cranial, however, they're not making enough ADH. So that's a pituitary problem usually. And this can be either from head trauma, so sometimes, as an effect of trauma to the brain, TBIs, and that sort of thing, there could become a pituitary issue. Most of the time, though, it's caused by a tumor. So a tumor, specifically a pituitary tumor, is going to cause the body to not make enough ADH. Let's end by talking about a few drugs. So the first one is furosemide. So you might remember this one from the lab, or if you haven't done week 3 lab, stay tuned. Furosemide is coming up. It's otherwise known as Lasix. This is a loop diuretic. And so there are different classes of diuretics. Diuretic meaning high volumes of urine. So whenever you see diuretic, remember it's the opposite of antidiuretic hormone. Decreases P. Diuretic, you're going to diuresis, you're going to pee a lot. So loop diuretics are used to treat either high blood pressure or fluid buildup. So again, we want to think about blood pressure and its relationship to blood volume. So if we have high blood pressure, either we have a lot of blood flowing through our vessels, we have high blood volume. And so we want to move that fluid somewhere. And the best way to do it, the easiest way to do it, is to increase the amount that you urinate in terms of water. So you want to take the fluid from the bloodstream and push it into the kidneys and have the person pee it out. Or if you have high fluid buildup, you've got a lot of edema, you want to do the same thing. You want to get that fluid out of the body and pass it into the urine. And the way that Lasix does this is by blocking the NKC, the sodium potassium 2 chloride co-transporter in the ascending loop of Henle. So it blocks that transporter, and so what happens, if you look at this picture here, is that in the luminal side, you're not going to get as much sodium moving into the epithelial and back into the blood. And so that means that the sodium is going to stay in the urine, and that what that does is it decreases the osmolarity of the medulla. So think about that osmotic gradient and the different components that build that osmotic gradient, right? We've got urea, we've got the salts, and then water will eventually follow and be taken up. Well, if we've just got the urea, we don't have the sodium, we don't have the salts moving into that interstitial fluid, we're going to decrease the osmolarity of the medulla. And by doing that, we're decreasing the driving force for water. So remember, water is going to follow that salt. And if you're not moving as much salt, you're not going to move as much water, which means both the salt and the water is going to stay in the urine, and we're going to pee more. We're going to have higher volumes of urine, and that will mean that fluid will be moving more out of the blood into the urine, which is great if that's the goal. If the goal is to decrease fluid. Okay. Another diuretic works in a different spot. So HCTZ, hydrochlorothalazine, this works in the distal tubule, and it blocks that sodium chloride symporter. And so there are a bunch of different types of thiazides that act here to have that same effect. And again, if we're looking in the luminal side versus in the blood side, if we're blocking the movement of sodium and chloride into the epithelial cell, we're also having that same effect of not moving the salt, and the water is going to not follow. And so we call that driving force of water following salt, we call that solute drag. So solute drag is when we move salt and then water follows. And so if we're decreasing that osmolarity, we're lowering that solute drag, we're going to lower the amount of water that leaves the filtrate, and we're going to increase the amount of water then that stays in the urine. And again, this is also used to treat blood pressure high blood pressure and to treat fluid buildup. Okay, and so this is showing you a picture of other types of diuretics that you might talk about or might hear about. So we covered Lasix, a loop diuretic, and thiazides, which act here in the distal tubule. You also have different diuretics that can act in the proximal tubule to block the transport of water, so osmotic diuretics. We didn't talk about carbonic anhydrase. We didn't talk about pH. I decided to skip that whole section about the kidney and its role in pH. So the chemistry here might not make as much sense, but there are inhibitors that work in the proximal tubule as well. And then in the collecting duct, we have antagonists to the effects of both aldosterone and to ADH. And so we have things that can work against the aquaporins or against the effect of ADH has on the aquaporins. And then we have drugs that work and block some of the downstream targets of aldosterone as well. So there are multiple different drugs that can act in the kidney. And then there are drugs that can act in the RAS system in general that will impact things upstream of what happens in the kidney. So you have ACE inhibitors, which block the production of angiotensin II. And then NSAIDs can work to help vasodilate things as opposed to vasoconstrict. So those are non-steroidal anti-inflammatories. Things like Tylenol have that vasodilation effect. Okay. So with that, we have finished the unit, which I know is kind of crazy. I'm going to open up a poll for you guys that I want you to answer. Users of this system. Okay, I think I opened a poll. Please let me know in the chat if you don't see a poll pop up on your little screen at the bottom. Basically, it's what do you want to do in class Wednesday? So my original plan for in class was to give you guys a case study to work on in class. But I don't want to add more to your burden of things you have to do. And so for me, I don't want to add more to your burden. I know you just finished that paper, you've got an exam on Friday, and I really don't want to take away from you studying for your exam by giving you more work to do. However, a case study actually does like reinforce some concepts. So we could do that. The other thing is to do a review session, but for that you would have to submit questions ahead of time. So I could open something up on Canvas where you would put in questions you have or things you want me to go back over, and I could do that on Wednesday. Or if you have another idea, that's where you click other and then you give me an idea, because again, I don't want to give you guys more work to do. I think that is not the right call. Also, as like professors are human beings as well. It's been a hard week for me. My grandfather has COVID. He is in a nursing home in New Jersey. And so he tested positive late last week, which has led to, it's just a lot. It's a lot going on. So I don't have it in me to do a lot more grading. Just like mentally, I don't have that energy to do a lot more grading. So I just want to try and find, something that is going to work for everyone. Okay, so it seems like most people want to do a question and a review session. So that's what we'll do. I will open up something on Canvas for you guys to put questions, topics to consider. Please try and make them as specific as possible. What I mean by that is please don't say something like, could you go back over the kidney? I don't know what to make of that, like a specific question, and I will go back through that and we'll kind of review material that way. Okay, I'm going to stop the recording.